So first, I'd like to do a welcome to country. Um, ALK Positive Australia acknowledges and respects the traditional owners as the original custodians of Australia's lands and waters, and their unique ability to care for country and their deep spiritual connection to it. We honour elders, past and present, whose knowledge and wisdom ensure the continuation of culture and traditional practices. ALK Positive Australia is committed to generally partnering with traditional owners and the Aboriginal community to progress their aspirations. I wanted to just give you a little bit of an introduction to ALK Positive Australia as well. This is the first webinar that we've ever hosted as a charity. We're quite a young charity. Um, ALK Positive Australia is a patient-led charity that's dedicated to improving the lives of Australians diagnosed with ALK Positive Lung Cancer. We have three core pillars, which are education, advocacy, and research. So this webinar is trying to address the first one of those, which is education, trying to understand ALK positive cancer and how the research that goes into trying to treat us. As an organisation, we are trying to primarily raise funds to support innovative clinical trials like the one that we're going to hear about tonight, and groundbreaking studies with the ultimate goal of turning ALK positive cancer into a manageable condition. A key achievement in that is the partnership that we have with the Thoracic Oncology Group of Australia, otherwise known as TOGA, um, to fund an ALK specific research grant which supports pioneering work aimed at improving treatment outcomes for us all. So that research grant, it's a $50,000 research grant that is being put out to fund fundamental research into ALK positive cancer. So we're about to get into it. I'd like to give a brief introduction to Dr. Malinda Itchins. She's an incredible Australian doctor. Um, she's a distinguished thoracic oncologist and a researcher based at Royal North Shore Hospital and the Chris O'Brien Lifehouse in Sydney. She has dedicated her career to advancing lung cancer treatment. She specialises in managing non-small cell lung cancer, particularly in ALK positive patients. Dr. Itchins has led numerous groundbreaking clinical trials and research initiatives aimed at developing innovative patient-centered treatment and strategies to improve outcomes. She is a leader in the Thoracic Oncology Group of Australia, Clinical Trials Group, and chair of the Lung Cancer for the Clinical Oncology Group of Australasia. Dr. Itchins is known for her inclusive approach to clinical trial design, where she actively involves consumers and patient advocates in the development process. And another one of her trials that she's been involved in, DYNAMALK, is an amazing example of that. And hopefully we'll get a chance to quickly talk about DYNAMALK at the end as well. This commitment to collaboration ensures that her research reflects the real-world needs of all of the patients, including those with rare cancers, setting a standard for how to integrate patient perspectives into clinical trial care. Her dedication to personalised cancer care and patient inclusivity makes her a role model in the field of thoracic oncology and ALK positive cancer research. She's an amazing doctor. She gives her time generously to all of us, all of the time. We're really fortunate to have her here. I'm now going to hand over to Dr. Malinda Itchins to run through um, the trial that she's here to talk to us about. Thank you, Dr. Itchins.
Thank you. Thanks, John. I'm not sure all that was true, but it is actually a privilege to be here. I didn't realise this was the first of the official webinar series, so I'll try and lift my game. But lovely to see everyone, obviously. And as it's being recorded, I said to John prior, I'll try to be somewhat official. I sometimes struggle with that as a concept. But, thank you for having me. Similarly, IT and I are not great friends, so John will kindly advance the slides for us. Now, I am mindful we're a group perhaps of differing scientific backgrounds. And this is actually the talk we gave recently at our annual scientific meeting with Toga. So apologies, but of course, the way that I talk through the slides on this particular study, I will try and frame in a way that doesn't sort of put us to sleep at 7 o'clock at night. So ALKTERNATE is the study. When you think of a clinical trial, the fun part is coming up with an acronym. So I spent a couple of days thinking about that one. But so this is a pilot study that we've run in Australia over a number of years, actually, and came up with the various challenges that is doing investigator-initiated research. And I think, as John has said, which is so important, collaborating with all of the key stakeholders, first and foremost, the consumers. So I will give a little bit of a sneak peek into some challenges, but I think you don't learn if everything is smooth. So it has been a journey, as they say. But this is a pilot study, which basically means it didn't need lots of people, but it was looking into a new concept, or a bit of a different hypothesis, as we say, as to how we might change the way we think about treating conditions such as ALK positive lung cancers. So, and now we can go to our next slide. So when I say hypothesis, I mean, the idea of this was born actually in 2016. So you perhaps all know that ALK wasn't even 10 years old then. But we were understanding that crizotinib was working. So the first drug we used, but maybe not but much better than chemo. So we were very excited and hopeful, but still the cancers were growing at a point in time, and lots of biopsies were happening, and we were understanding that, each of the cancer cells within, the lung cancer or the secondary sites that were biopsied, they could differ a bit. And so, the idea is that you give a tablet and it works well, and it might work on a lot or all of the cancer or some of the cancer, but it's not sort of a one size fits all, perhaps for some, and perhaps through time. And how long could that be? It could be months, it could be years, obviously, hopefully it's decades. Some of those cells work out ways to kind of, outsmart the tablet. And that could be for various reasons. It could be because a mutation or a gene change happens where the drug can't bind within the cell, within the cancer cell, because that's what these tablets do. Or it could be that actually something that's non-ALK-y starts to actually emerge and tells the cells to grow for another reason, and the tablet's not actually targeting those pathways. So the idea is maybe we could alter what we're throwing at the cancer to trick it, to maybe prevent the drug resistance, so delay it even longer, or change it to be more manageable. So the idea was obviously quite what we would say novel, or a bit outside the square. But what was really important about this study when we first designed it was we were starting to see liquid biopsies, and you all know that perhaps there's been quite a lot of runs now in that space and in advances since then. But we wanted to have a look at how much we could find from the blood, because the idea of a tissue biopsy can be, confronting, potentially have some side effects. As well as we don't do them every other month, because that would be cruel. So what can we do in the blood to see how things are going with the cancer treatment, to inform, just the best care? Does it value add? Next slide. So actually, there wasn't a whole lot of background material to support our hypothesis, just that deep gut feeling. But there was a couple of, so Alice Shaw might be somebody that you've, has a guess, heard of. So she published, actually, around the time that we thought about this concept, a super important clinical trial of one case, which basically really made us all think. So that's that diagram to the left there, which is basically an individual who went on a number of different treatments. And so it's like a calendar through time for that person. And what happened with the gene changes, outside of just the ALK gene in tissue biopsies. And what was super interesting, actually, is that individual had a number of different treatments, started on crizotinib, as would be usual, and thankfully, because Alice Shaw was looking after them, got onto lorlatinib before it was sort of had a proper name, and then actually developed resistance to the lorlatinib. But what happened at the time of the biopsy after the lorlatinib wasn't working as well is that individual had some mutations or changes in their cancer that actually resensitized them to the crizotinib. And actually they went on to the crizotinib again and responded. So that's obviously pretty cool. And the idea of getting as many runs out of any medicine is a no-brainer. And that's not something we really ever thought about in cancer so much. I mean, there are some chemos we might use again in time, but we never really thought, we usually think when you're resistant to it, you're resistant. You know, we maybe wouldn't use it again. So that was kind of like to us, wow, you know, things are really dynamic because, you know, until recently as well, it's the biopsy at diagnosis that guides actually all the treatments, especially in other cancers. I think ALK's really changed the landscape also for people with other non-ALK positive cancers that, the postcode of what's going on in the cancer 4 years down the track is quite different. So, you know, we want to know that. We don't want to just be speculating. So, and then to the right was a really important study as well, with Elizabeth McCoach, who does a lot of really important and very scientific work for ALK positive cancers. And this is an individual that had liquid biopsies through time. So this was perhaps the first similar type of report that said, well, if you do liquid blood tests, you can probably see there. So basically the colors and the ways they bubble is how much of the DNA is in the blood. And the color is which gene that is that's cancerous. And you can see that it sort of selects out where it says responding, and maybe it's not really detectable in the blood, but maybe then it starts to come back again a bit before progression. And, you know, maybe through time we could track that and adapt what we do with treatments, you know, based on the blood test. And, you know, that's also a concept that's still being developed, but obviously a very enticing idea. So this particular study used 2 ALK inhibitors, which I'll show you on the next slide, the design. But what it did was it alternated them. So you stayed on, and lorlatinib was the important one in this one, and I'll show you again in the design. But the one that you had small bursts of was crizotinib. For various reasons. And I would say, you know, don't forget this was designed in 2016. And I often say, like, ALK is like dog years, like a year in what changes is like 7 or 8 years for other cancers. So, you know, this is basically the dark ages when we designed, you know, this particular study. But what was a bit interesting is when we started is actually lorlatinib, we were getting a bit of information as to, you know, in those that were taking it, why was it stopping working? And there was another pathway that was, you know, in about 1 in 5 people going, you know, starting to misbehave. And that pathway is called MET. So ALK is one type of protein, you know, MET is another. And actually crizotinib wasn't first designed to be an ALK inhibitor, which is a bit interesting. They accidentally realized that it worked for ALK lung cancers. It was first designed to be a MET inhibitor. So we thought, oh gosh, well, that's a bit of a crazy coincidence, and we'll take that, thank you. So crizotinib might, in this particular study, for example, prevent that MET pathway, you know, from popping. So again, that evolved as we went on. So next slide, John. So this is the design. We don't have to, I guess, spend too much time on it, but basically, so it's a single arm study. So everyone that came on got the same treatment because it's a pilot. So we wanted maybe a couple of dozen people. And we wanted it, as John said at the start, to be pretty inclusive. So you could have had any number of prior lines of treatment, provided that the most recent wasn't crizotinib because you're about to get exposed to that again. And that you had a second generation ALK inhibitor in the past, an alectinib, a brigatinib, a ceritinib, just because we wanted to make sure that, you know, this study was coming in at the space that was looking at that resistance, you know, question. So what you got is 3 months of lorlatinib. So you got your induction basically. And, you know, we did actually know that lorlatinib might not work for everyone. And we didn't do any sort of other tests to check if, you know, the liquid blood test, for example, didn't say whether you can come onto the study or not. Everyone could come on. But it wasn't until we saw that the lorlatinib was at least helping with the cancer, so at least the cancer was stable, that we would then flip onto that alternating treatment, which included then a month of crizotinib, 2 months of lorlatinib, because lorlatinib is the one doing the heavy lifting. The crizotinib was there actually not so much to keep shrinking the cancer, but it was to help to maintain the sensitivity of the lorlatinib. That's what we sort of wanted it to do. When we first presented this actually to a group of doctors, they kind of said, and it was happening a bit in the melanoma space, that maybe you could have a holiday altogether from the medicine, you know, so just take a break. We did a survey at that time, and I don't know if anybody in this particular group was involved, but at that time that made people very anxious to stop medicine altogether, and we were still learning on the job. I think maybe there's been a little bit more interest in that in the last year or two in ALK and EGFR, and maybe having short, even breaks from the treatment. But so the crizotinib we kept in there, but we kept it in a skinnier interval, so a month and then back onto 2 months of lorlatinib. So we wanted to test a whole host of things, because this is actually what you call a feasibility study. So can we do it? Do patients just say, up yours, I don't like this? Or are they actually invested in it? Are they feeling okay? We checked drug levels in the blood to see that, we didn't make the blood levels of drug too high or low that might be dangerous. So we really wanted to check that it was safe, that people's quality of life was okay, and is it actually potentially working? So what we defined that was, well, we didn't know, but I guess it would be based on the original evidence we saw with lorlatinib on its own, which was really if you'd had alectinib prior, that we say the median or the middle amount of time that people might stay on it was about 6 months. So we knew we need to do better than that. But that was kind of our benchmark. So lots of different tests to see if this looked like it was feasible. And then next slide. So this is a bit of a timeline, which I've already alluded to, but it's really brings a lot of respect to those that do big randomized studies, because it takes quite a lot of time. It took a number of years actually to even negotiate Pfizer, perhaps providing us the drug and then some funds. The budget was a bit skinny, and even to get through the local ethics took about a year for lots of reasons. So we opened in 2 sites. We would have liked to open in many for obvious reasons, but that gets really expensive. So we decided Peter Mac and North Shore, and obviously Peter Mac. We had expected to bring lots of people on, but then the pandemic came and they weren't allowed to actually put people on the study for nearly 2 years. So that created a few grey hairs. So, 1 out of 2 sites then, for various, important, worldwide reasons, not being able to recruit is tricky. But we overcame those hurdles. I think the main thing is we're all kind of solutions-focused people, I would hazard a guess, also on this call. So we eventually got there a number of years later. We recruited, or we screened to put on the study, 20 people. Of course, it would have been nice to have some more, but those are the challenges. Lorlatinib also, thankfully, for the greater group, went on to Medicare. And of course, when that happens, people want to stay. You maybe have been with your oncologist for years. I completely respect that it is, that relationship is very therapeutic. So perhaps, going across town or even across states for a study is a, a tall ask. But of course it was an opportunity, and some people did do that, which, we're obviously very grateful for. So, and believing in wanting to test the science. So what I didn't mention is if the cancer wasn't, agreeing with the alternating therapy, you could change over to go on to continuous lorlatinib. So we sort of built that one in as a safety measure too. And we did lots of MRI brains. So because crizotinib doesn't get into the brain perhaps as well, and definitely not as well as the newer ALK treatments, and that was all evolving as we went. So we were rock solid on the MRI brains. We really had to make sure that we were keeping people safe as well. Some people don't like MRI brains though, so that also brings that challenge. So next slide. So as I said, we screened 21 people. For various reasons, people didn't come on, but an important reason was actually there were 2 patients where the liquid assay actually confirms that they had a different cancer process that wasn't ALK positive. So what I like about that is we quite importantly didn't put them onto the study because it wasn't the right study for them. And one of them actually I'm now treating nearly 5 years later on immunotherapy, and they were actually an individual that had a false positive ALK test from the outset, and their FISH test, although it was positive, didn't transpire to be ALK positive. So I like that the liquid can also help us to make sure we're selecting the right, treatment from the outset, because you all know that that's so important. So we enrolled 15 onto the study. So proof of concept. So, wanting to see that we can then start getting people onto alternating. Unfortunately, in 3 people, they didn't respond to the lorlatinib. So we did biopsies on those people. We worked out, the reasons why, and we tried to get them rapidly then onto another effective treatment. So 12 people went on to do the alternating treatment. And during the few years on the study, 10 of those people had growth of their cancer and stopped alternating. 2 actually remain on alternating treatment. So it's continued to work. So, it has worked in some people for many, many years. I want to say 5 to 6, but now I'm getting confused by our start point. But 9 of the 10 did cross over to continuous lorlatinib. It worked for about 3 months, so it wasn't a long time when you went over to the continuous in that setting. So that was sort of the people that we brought in, and obviously at the end we can have some questions about what that looked like. But that's a bit of a glimpse at the cohort, as we say. Next slide. So this is called a swimmer plot. So each line is a person. Each colour is their treatment. Time 0 is when they came onto the clinical study. So why did we want to put this in? Well, actually, there were some people that had been 5 or 6 or longer years into their diagnosis. There were some people that had only been, 9 months into their diagnosis and only had alectinib. So there was a really, really broad group of people in the previous treatments they'd had. And that comes actually a little bit then down to the findings, because we're doing a one-size-fits-all treatment to a group of people that have come from very different treatment backgrounds and durations of benefit. So the red is how long they were on the alternating treatment. Down the bottom is in years. So, thankfully you can see someone nudging out there to 5 years. And if the arrow is there, they're still going. But some people, of course, we did lose while we were on this study. And that's where the fire really burns of how, we do better. Some people had the opportunity to go into the newer ALK 4th generation ALK inhibitors. So another study opened, another opportunity for them. But lots of different treatments, as you can see, cycled there. So next slide. So we love this in oncology, but these are Kaplan-Meier plots. Some of you on the line, I know them well. But basically they're everybody that goes onto the study and what happened through time. So with, for example, survival. So the primary outcome of this study was what we called time to treatment failure. We called it that because you might stop because it was a bit toxic alternating, you might stop because you didn't really like it, or you might stop because your cancer grew. So that's kind of what you call quite a pragmatic or kind of a real world outcome. Like this was a clinical trial that was more for the problems we're having in the clinic. So time to treatment failure, for those that went on to alternating treatment, the median, we say medians, and I know that it's a bit of a strange term, but it's not the average, it's the person in the middle. So it was 11 months. So it wasn't obviously as long as we would like. I mean, I don't think we should ever accept. We always want more. But it thankfully was competitive with what we're seeing. And there were some signals that actually some people did very well. And we know some people also do very well with lorlatinib, on its own as a continuous treatment. So those that came on, the median that, after they'd had their various previous treatments and came onto our study, the survival was nearly 2 years. The progression-free survival, so the time before the cancer grew in everyone that came on, including those that the lorlatinib never controlled the cancer, was 6 months, about the same as what it was for the study that Ben Solomon read out. But then we wanted to look, obviously, at the extra information we got about the blood. And what else can we explore? So like everything, maybe the first thing you go fishing for, actually you start looking, and the devil's in the detail, or hopefully the promising information to move forward comes from the detail. So one of them is actually, the blood test showed that, there's a lot going on apart from the ALK positive gene for many people. And so in those that may have had, when they came on, another gene mutation in the blood called TP53 that gets a lot of airtime, I think, in our space, is actually, if you didn't have that TP53, you went on for a long time, 27 months. But if you did have it, 5.5 months. So maybe we need to think about, do we do things differently if you have one of those other genes apart from the ALK? Similarly, if you had no detectable DNA in the blood at diagnosis or at 6 months. So we did it at diagnosis, every 3 months, out to 6 months. So if it was clear, certainly by the 6-month mark, versus if it wasn't, there was a difference. So really in those where it was clear, it was sort of 25 months on the treatment. But if it wasn't, it was less. It was 8 months. So again, it immediately speaks to us that if you're not clearing the detectable, tumor contents or DNA from the blood test, well, maybe we need to think about that differently as well. And what else can we do, to be more proactive at that time point? And I think that's where I really started focusing, my attention. This study didn't do any of the doing. It did the exploring. So yes, there are a whole host of things we're thinking about at the time. Should we give radiation? Should we add another drug? Should we? And that's what we'll talk about in the final slide. But absolutely, it's those that don't perform as well. We need to be lifting the bar. So next slide. So this is a busy slide. I'll talk, just bear with me, but I'll talk you through it. So the reason I wanted to put this up is really everybody was very different. Everybody was very unique. The signature, or what we were finding in the blood, it's just to me, ALK positive. Thankfully the medicines work for everyone, for a long, long time, but there's really a lot of intricate differences within each, individual that we had on this study. And I think we're seeing that more and more when we look deeper. So for example, there were lots of different types of, so we know ALK and ALK rearrangement, and the ALK fuses to another gene. The other gene it fuses to is most commonly EML4, but it can fuse in different ways to EML4. So, and there's two variants we talk about a lot. There's variant 1, which is the most common. There's variant 3, that's the next most common. The reason I bring that up is actually we were seeing some evidence that maybe lorlatinib works better for variant 3, if you use it, after prior ALK inhibitors. We're still trying to understand that. At the moment, the type of variant people have, well, first of all, we weren't routinely testing it because we didn't get that from the FISH test. But now we're doing, more fancy gene testing routinely now in tissue. We are getting that information. But at the moment, it doesn't change what we do. But we're still a little bit open to understanding could it in the future. So that's why it's important we tease out the differences with the different types of ALK fusions you have. And actually here we had 6 different types of ALK fusions, including, somebody that had a rare non-EML4 ALK fusion. Now, then there's the ALK resistance mutations. So as some of you will know, some of you will not know, but there are different mutations that basically develop, or changes in the genes that stop the drug binding, as I mentioned earlier. So stop it being able to get there and bind and shut the cell down. And there's a whole host of different ones. Actually, there's dozens. And a common one, for example, for earlier ALK inhibitors not working is called G1202R. Everything's a bit of a code. But for example, we did see that in this particular study, and that's the larger, so the second pie down, the larger greenery there is G1202R. But you'll see that's not the whole pie, is it? So actually there were 18 different ALK mutations, and one person, for example, had about 5 different ones. So it's starting to get, very, very unique. And then below that, though, a lot of people didn't just have an ALK mutation. They may have had other pathways, other parts of the cell pathways working harder. So, of course, how else can we look at those pathways? And, as I said, TP53, so the bottom pie, the bigger green part, was the most common. There's a whole host of other ones there, including some MET, including some EGFR, but different types of EGFR, like amplification or more of that particular protein. But what was important and definitely not found is nobody on alternating looked like they had resistance to lorlatinib because of the MET pathway, starting to fire off. So that was something we expected to see, and that is something that was not found. So we didn't see MET. So we seem to have altered that process. And then each of these plots in the middle are a different patient again, and a little like the one I showed you at the start. And actually, because we wanted to kind of re-demonstrate what we saw at the start from those case reports, and having a look at what we find at diagnosis, what hopefully selects out to not be seen anymore, and then what changes. So we definitely did see that changes were happening in the blood, before there were changes on the scan. And as we've heard a little bit, for those of you I know who attend the scientific meetings, is actually that was happening with some lead time, maybe 3 to 6 months. So maybe there were changes we were starting to, well, there were in this particular small study in the blood before we saw changes on the scans, and certainly before we saw any symptoms, from the cancer growing. So it was interesting. And for example, the one in the middle, that individual is still going. So look, and there was no detectable changes in the blood, forever whilst on the study. So that is the goal. And others had differing changes. So down on then finally on that right side, is each of the colors are different gene changes. And if it starts with ALK, it is a type of ALK mutation. But as you can see, and as I mentioned, sometimes it's the ALK mutation with other non-ALK mutations. And I think that's really now also that we're using lorlatinib first for a lot of people with a diagnosis of ALK positive cancers from 2024 onward. That's going to be where we put our attention, because it's working so well on the ALK pathway. How do we keep everything else in order? So next slide. So just a really brief proteomic, it's a fancy word to say. So you can look at genes and you can look at proteins. Genes actually, code what a protein is, but it's the protein that does the things to the cell. So looking at proteins in blood is a bit of a newer thing to do. So we talk about gene tests, but we don't really routinely do proteomics. So we wanted to take a look at the blood and see that we could do it. And so we engaged an expert in protein proteomics who does a lot more with tissue, but he went in with blood with us. And we found actually a whole battery of different proteins that were different in the people that went onto the study, actually nearly 400 proteins. So that's a lot. So then we've got to wrap our head around that. So we tried to then, cluster, so we call them signatures or different types of groups of proteins into what they could mean. There's so many things we can keep looking at from here, but just as an example, so the red and the green, don't worry, I don't really understand it either. But what I would say is that actually the protein changes again in each individual were a bit unique. So there were different protein signatures in each individual, and they kind of did trend the same. So you stayed sort of in your lane throughout. The other thing was, you may have seen in the past that people, so with lung cancer and ALK positive lung cancers can develop blood clots. And we're still also understanding that, of course, and then so that we can prevent it. But what is it and why? And I mean, we did find, for example, through time on this study, if a whole lot of different proteins involved in clotting changed, actually the survival wasn't as long, or the time on the study wasn't as long. So could there be other signatures or things we look in the blood beyond genes and mutations like proteins that could help us to understand, how we can optimize treatment? So that's kind of what this, and it's, as you said, you're supporting ongoing research, which is amazing. And maybe we could start looking beyond the genes. And this is the type of work that could be done. So next slide. So, and just finally, and then of course, how was it tolerated? So I think, thankfully, really not many people at all had a serious side effect, which means it was bad enough to maybe bring them into hospital or interfered certainly with their quality of life. Although when you say grade 3 and 4, it does say there that 60%, so 2 out of 3 may have had a grade 3. But to be honest, as you might know, with lorlatinib, these can be what we call paper toxicities or blood test changes, like the cholesterol goes zing and goes really high, or weight gain is definitely an issue. So that can be a grade 3, but maybe, although of course that can affect quality of life, the weight gain, not the type that is unsafe necessarily. So definitely people on lorlatinib. So 27% or 4 individuals did need a dose reduction for helping with mood, and that was important, and it really helped. And crizotinib, in one individual caused relatively significant esophagitis, and we had to change our protocol then and remind everyone to take it with food, as you may know, because that person wasn't, and the tablets were actually getting stuck in his food pipe. So they got fished out. And then we did fancy blood tests for drug levels, and that was interesting actually, because we were a big budget to do that, and we were doing them in China, and actually China decided that they wouldn't accept our samples if we didn't have a negative COVID test on everybody when they were taken. And anybody that's had to do blood level testing on a clinical trial knows that you hang around a long time and you maybe eat bad hospital sandwiches and you even come back the next day. So that made me feel very sick inside that we couldn't, it took a long time to get them then processed. But eventually China opened up the borders to accepting blood tests because the pandemic still had ripple effects even until this year, with regards to also sending tissue samples to other countries. So actually, I was happy with quality of life. So we benchmarked it on quality of life for a normal, I don't know what normal is, individual, but it sat nicely there. Obviously some people dipped, but it actually also improved on the study. So the trends were reassuring. What is important though is 50% of people did have some progression in the brain. As I said, we did MRI brains at least 3 monthly, even more frequently at the start. They all bar one had some cancer in the brain at the start when they came onto the study. They were managed with stereotactic radiosurgery, and at that time went over to the continuous lorlatinib. So we tried to be super obviously careful there. But of course, it would have been nice to have 2 brain penetrant ALK inhibitors, and that would be one of the next important steps. I would say, and we didn't have a way to measure this, and it's a shame, but people anecdotally did share they felt a bit better when they had a break from lorlatinib. So, I guess a little holiday from medicines can, not only for mood effects, but different types of medicines, other ones we use cause, potentially lower limb swelling, etc. Can be nice to have a break. So we did some tests on memory. They were a bit simple, to be honest. Most people remembered them by sort of the second go around. But I would say that most people didn't score full marks. So we were mindful that, we have to be very careful about also with lorlatinib, helping support people with their cognition. So next slide. So to conclude, I think, there was a signal that we can do this, that it was safe, that we kept people's quality of life. We didn't compromise it. Really, the survival, as you saw, was comparable. Some people did very well. But what was important was that, doing regular blood tests to look at the liquid and what was going on with the biology of the cancer or the mutations actually was quite informative. And so I think that that's really the grain of gold we take forward. And so, yes, we can absolutely expand this type of approach in different ways. Doesn't have to be drug and drug, might be drug and radiation, there's different ways, and we can discuss that now. I'm nearly about to stop talking. Of course, do we do it in first line? That will be a big plunge, as we've seen some very encouraging lorlatinib data, and obviously alectinib and brigatinib data prior to that. So, or do we sort of alter things, when there's a change in the blood? So I think that's what the next slide just is, and there's a brief look at just my wish list. So this is small, but as we've alluded to, there's a whole lot of different medicines that can help. Anti-angiogenic ones that block blood vessels, they work potentially for TP53. There's other targeted therapies, an EGFR inhibitor, chemo, radio. PET scans, I think, would be interesting to do with liquid. And then look at not being fixed, not being 2 months, 1 month, 2 months, maybe being keep going if there's nothing detectable in your liquid. But maybe if it changes, could you do 2 medicines together? And then perhaps when things clear, you might sort of decelerate the treatment. So sort of looking at accelerating and decelerating, based on, what's going on with, particularly the liquid blood tests, I think would be quite an interesting next step. And we published this type of a concept last year. So, and then final slide, I hope. Let's just have, no, the little one. Yep. So we've got the publication, which I shared with you. And, obviously, please have a look as it details the experience and the findings with the study and future directions, of course. Next slide. So obviously, huge thank you to the patients that went on. Super inspired by everybody that got onto the study. We did really rely on people to refer from other centres. So all of the doctors here referred to patients. So that was excellent because to do studies in rare cancers is challenging. So we were grateful to Pfizer. We had a really nice liquid assay through the US with a research partnership there. A really good assay that's been very validated for ALK. It really picks up the fusion, which is important. We got some nice philanthropic donations as well. And obviously, I think, but I'm not allowed on the socials, there was also great support from the ALK Positive consumer group and through your channels, which we're hugely appreciative of. And so the final one is actually, we can probably skip through it, but these are some emotions that come from this type of research. Some good ones, but there's a whole host of different challenges, and also, but you just got to focus on believing in, the science and the passion, and obviously you've got to follow through for the people and the patients that invested their time. But woo, it's you can see why some studies don't make it. So yes, so I guess we want to spend some time talking now, I hope. But that was an overview of the experience with ALKTERNATE. So obviously very happy to discuss now.
Take a breath, Malinda. Have a quick drink of water. And I'll ask Amanda. I saw some questions pop up through the chat. I'll ask Amanda to open up a chat and maybe ask you some of the questions that came through the chat, and then we'll move on to people who have got some, who are on live to ask further questions. But we still want to try and maybe find 5 minutes at the end to quickly run through DYNAMALK as well, because there's people here who would be very interested to know about DYNAMALK as well. So do you have some questions for us, Amanda?
I do. I'm going to kick off with a question that was emailed through from Graham, and then we've got some questions from people that are online now. So I'll pass to them to have that conversation. But one of the questions that Graham sent through was, do you think that the trial could have been done with a holiday and then a TKI? So rather have lorlatinib and then no second TKI.
Yeah. Well, hindsight is always, a little easier, because I think now we see lorlatinib work really well, really quickly, really deep responses, and I would feel more comfortable with that concept. I guess then you would have to make a scientific, rational call on how long those holidays are. Maybe it wouldn't be a month. But that's certainly something that has been discussed a little bit more in the last couple of years, and I like that as a general way of doing things. So there's another drug I'll just briefly elaborate. So for MET exon 14 skip mutation positive lung cancers, I mean, it's a bit of a mouthful, but they got a medicine on PBS last year, tepotinib, like teapot, and actually you do get really bad swelling, and we have to do holidays on that medicine. And I think we might learn a bit because diuretics and things don't really work, and so I think we might get a bit of information from that. But the evidence I've seen thus far actually is if you're having a holiday, in that circumstance, obviously for a side effect, which is a little different, we don't see compromised cancer control. So very, very interested to think about that way of doing things more, and obviously research it formally.
Excellent. Thank you. There's a couple of questions here from Alan. Alan, would you like, I can ask them for you if you'd like. So the one question that he had was, you mentioned early on in the piece about that people dropped off the trial because of sensitivity to lorlatinib. Like what do you mean by that? What does that mean?
Oh, did I use those words? No, I think I meant at the start. I did use this word. Oh, whoops. Yes. Sorry. 3 people didn't continue on to alternating because the cancer wasn't at least stable after 3 months. So they were enrolled in the study, they started, but we did not think the right thing to do was then a new type of treatment approach if the cancer wasn't being controlled, from the first, experience with lorlatinib. The step above them, when you say dropping off, was so there were 2 that didn't have, as I said, the ALK positive. The other reasons before then coming onto the study, one person just decided they didn't want to, and we respect that. One didn't have measurable cancer, because to go into a clinical trial, you need a blob, that you can measure. So that's a good problem to be having. And the others' blood tests stayed a bit skew with. So we do need, for a clinical trial, things to look at the baseline, all quite safe and happy. So sensitive to lorlatinib, otherwise it was the dose reduction. So 4 people did need, because of having some, we say neurocognitive, or some of the side effects of thinking and mood. So that was where the sensitive perhaps is what I meant.
Excellent. And Alan also asked, with regards to the study, are all the public hospitals in Australia aware of it? How, is that information shared openly?
Oh, we try our best. Look, I don't want to say too much about public healthcare. That's for another webinar. But look, to say there's cohesive communication would be a stretch. But I think in the lung cancer treating community, we're pretty cohesive, and our communication channels are quite good. But as you know, there are people being treated with ALK or any type of rare cancer anywhere, and there perhaps some people do inform me they've found out more about this through your forums and channels, as opposed to their doctor, and that's okay. I mean, this is all happening quite quickly, these types of changes. So yeah, we communicate, but we could always communicate better.
Toga does a pretty good job of communicating the trials that it's involved in as well, with other clinicians as well.
Yeah, exactly.
I've got another question that Graham sent in. I'll read that out.
Graham, he's on fire.
Oh, yeah, he sure is. I think he's in the sky at the moment, so they were sent in early, yeah. Yes. So the question is, did the dose, onboarding and offboarding to lorlatinib lead to any increases in adverse effects when compared with Crown data to, straight
Good question. So sorry, what I didn't mention is in a lot of that negotiation to start this medicine. So by the way, when we had the negotiations for this study, lorlatinib wasn't internationally, FDA approved yet. So they were pretty delicate with what they'd let us do with the drug, which is why we had to be very careful with the way we dosed it. And we had the Pfizer sort of pharmacokinetic, they're called, or basically the drug guru, let us know that to safely do this, we needed to stop the previous medicine, and I didn't bring this up, 48 hours before you go on to the next one. So to make sure that, one didn't boost too much of the other, or vice versa. So when we checked the drug levels in the blood, doing that, they stayed at, the concentration we'd expect, if you were taking the medicine continuously. So that was good. And then when we looked at the side effects, and, the caveat is always, this is a group of 12 that went on and alternated. But we did not see any side effect signals that made it look like we were adding toxicity with doing this alternating. To be honest, I think we were maybe helping a little with it, but again, I've got to remember that I'm, quite invested at this point, and it's all, speculation in the sense that talking to the people on it, as I said, when they had a break from lorlatinib, they did feel a bit clearer. Many people were still working. And so then I guess the other way to answer that question is how many people needed dose reductions because of side effects. So they had to go down on their drug dose, and it was very comparable to, for example, Crown or previous, crizotinib studies. Very small numbers needed to go down, but if you needed to go down, that was obviously completely fine. So that is a big focus of changing the way we use medicines, and if it's starting and stopping, you've really got to make sure that you don't interfere with it being effective, and you have to make sure it's still well tolerated.
Excellent, thank you. There's a couple of questions here from Peter. So Peter, one of the questions, what treatment did the lorlatinib non-responders go to?
Yeah, Peter, that's a hard group. So they went to chemo. Oh, they went to, as in from the outset, they went to chemo. But sorry, one was didn't make it to more treatment. So he had quite a lot of the cancer already in his lungs, so he had to respond, if I'm honest. But we knew that he was going on to the treatment he would have gone on to, being lorlatinib. So it was very much during this time chemo. I didn't elaborate too much further because just talk about the tissue biopsies. So sorry, we did do tissue biopsies in those where it was safe. And for example, one who progressed on lorlatinib at the other side of the study actually transformed to a squamous type of lung cancer. So different. So we talk about small cell transformation, which is something that is thankfully rare, but can happen. But this one actually changed to squamous. So if I'm honest, there were some findings, and one final thing I would say is tissue is still important. So another man actually had progression in the brain, and we decided to do a resection of that brain tissue. And he had the ALK fusion with an NTRK fusion. So anybody who is following other genes in lung cancer would know NTRK occurs in 0.2% of lung cancers, but there's an extremely effective medicine now on Medicare called larotrectinib. So it works about as well as lorlatinib. And actually that person I can share is going on to larotrectinib with lorlatinib. Is that a standard thing to do? No. It's very personalized to him with some caveats. Is actually we couldn't find any evidence of that having been done before. But again, as I said, my main conclusion is every individual is very different. So we've got to keep an open mind if maybe that's, a pathway that could be helpful.
There are more questions, but I know that there are some people that are very keen to hear about DYNAMALK, and I'm conscious of time. So if you can give us a little bit about that, and then if we have time, maybe we can come back to some other questions.
Yeah, no problems. Lisa, do you want to ask your question?
Gail.
You knew when my camera went on, didn't you?
Okay. I was waiting.
Okay, so yeah, I love that concept of the NTRK person who is now combining the two. For people who are on this call, I suppose, how would you have that conversation with your doctor about those possibilities, since we know it's not standard of care approach?
Oh, I'm learning on the job. I would say, it's a lot of paperwork for us after hours. I can't even speak because, if your after hours involves kids and dependents and nappies, so because it's not standard and it is traversing the way we historically have done things, which is very cookie cutter. And it means maybe if it's not going through Medicare channels, well, you have to talk to multiple drug companies and they don't always like really crosstalk. So it's involved, but it's conversations we need to have, right? So so it takes more time. It takes us making sure there's an informed decision. So explaining the risks, the benefits, and the unknowns, and then deciding it's, because I've got another man, who's funding tepotinib, so that medicine I said, but with an ALK inhibitor. And I really want to say that this is currently, we don't know. We're learning as we go. So we're trying to just continue to, and, he's gone through standard of care treatments. That's another really important thing to say. So he's done the standard things and now paying out of his own pocket for the tepotinib, and it's really expensive. But it's a decision we made to do. And with early scanning, So when you do it, as you know, more blood tests, closer scans, and plan B's in place.
Yeah. So, I guess it's just not all doctors are sort of as open as what you are. And I know the challenges in the group have often been, that discussion with the doctor, even on some simple things, simple aspects of care, let alone trying to jump into those complex conversations of, multiple mutations. So some doctors will, some won't. I mean, I know someone in the EGFR space who was combining osimertinib with the MET inhibitor, and the first doctor said no. She brought a whole heap of case studies to the doctor and to another doctor and another oncologist, and they said yes. So sometimes
it's data to do that now. So there's actually some reasonable data to support it. Yeah, I hear you. I think look, we are thankfully in a digital space, and there are probably very good reasons also why doctors wait. There are good reasons for a high level of evidence, but there are challenges in rare cancers that, we all know here. So we have to reach a happy medium of going outside of the bounds of, perhaps where the evidence is or where it stops, but keeping it safe. And that is, if we're always doing that, I think that we're well justified. And when I brought up the digital space, because I think that you've got opportunities. As an oncologist, I'm often recommending to the people I look after to get another opinion, have another discussion. I think it's super healthy. And, there's different perspectives. So I think it can be healthy. Obviously, it's a lot of energy to talk to somebody else about everything that's been happening, but I highly encourage it. I think everybody does. I was just reading the Cancer Council lung cancer guidelines, and they suggest it in the actual guideline. So it's a second opinion might help to also keep those discussions flowing. DYNAMALK in a moment is, thank you, a study that kind of just flowed on from ALKTERNATE, of course. So, Lisa knows well, liquid is pretty encouraging and hopefully more accessible, but we're not quite there yet. I would say that there are now more rigorous discussions being had with government to try and get it more readily accessible, and hopefully it won't be too far off. So this is a study basically looking at anybody with an ALK positive lung cancer anywhere around the country who may be starting a new ALK inhibitor therapy, any line, that can have liquid assays, that we get the results back real time, and we can look at those results and see if there's any way it may value add to their treatment either now or later when we go back and look at the results. So as you can see there, and as we heard earlier, a really great collaboration with the ALK Positive group. Next slide. So we can probably keep going because we've sort of spoken about this. Through time, things change. And so this is, as I said, a multi-center study. So Aurora is a platform study we have to do real-world research across. I think we've got 30 hospitals on board now, but you can actually still go into the study if you're not at an Aurora site, if you dial into an Aurora center. So if your doctor gets onto the phone and with one of us and we do a quick sort of virtual consent to go on the study, and then we ship the kit. So we can ship the kit anywhere to take the bloods, that is. So we basically want to look at what changes over time. We're actually incorporating PET scans into this study to correlate them or have a look what the PET looks like when the blood test happens whilst monitoring the cancer treatment. So it's just an extra tool to see what's happening at the start, at the first scan, and then if the cancer grows, or 18 months down the track to see what's happening. So next slide. So the design is as I've described. I mean, it's anybody. We really do have a bit of a focus on those that are newly diagnosed. We want to get 30 people that are a new diagnosis and going on to lorlatinib, and 20, maybe more, depending on our budgets, that have had previous ALK inhibitors. So there's some information, and we've got the trial registered through the usual channels.
Are you struggling for anyone in particular, Malinda?
Oh, the new diagnoses are not easy for all the obvious reasons. There's so much going on in that space. But we do have at the moment, I think, 14. And I think we have maybe 18 pre-treated. So to be honest, I think we're doing pretty well. But at the same time, we've tried to have kits available in as many hospitals as possible so they can. So, for example, Peter Mac today did phone and put someone straight on. So, that's how we want it to be. And it can't interfere with you doing what you have to do with your standard treatment. You go straight onto the tablet, but if we can intercept with that blood test ASAP, that's the key. Over about 16 months is the plan. So as I've mentioned, the other extra, I guess, is that we've got a group of us that it's via email that we communicate, but we review the results. We feed them back to the referring clinician, and we give a bit of a synopsis or a summary of our interpretation and how it may be built into the context of that individual's treatment. What are you laughing at, John?
Oh, someone just popped up in the chat. That's me who joined today. So
Graham,
someone here joined your trial today.
Sorry, we are completely anonymous, so I can't see the chat. So don't worry, all of our ethics are rock solid. So thank you. So yes, next slide. And sorry, there are all this. We need better reach in Northern Territory. So please, if you have members from the Northern Territory, let's try and get them involved a little more as well. This is outdated, sorry, but this was our recruitment at the Toga ASM for the arms. And obviously at that meeting, we acknowledged, again, the reach of those that have referred people so far. So I think there's a good awareness. And the key is it's really not onerous. So thank you today. And it was a quick phone call, and it was a yes, and it was a do, and it's happening. So that's how we want it to be. And then I think there's just a final slide. And sorry, I'm noting it's 8 o'clock. But so basically, we're happy to receive emails from, individuals in the ALK community direct. So you can email us there, and I'm very happy these slides are shared if that can be helpful, if anybody reaches out to you. So that email will get you through to, to the blood test.
I think there was just one complicated question that Chris Henry had in the chat that I might have, if he gets to ask it himself, if he's available to.
Aloha.
Unmute yourself, Chris, and ask a question.
Yeah. Hello, everyone. Thanks a lot. Thanks for taking my question. Sorry if it came across complicated, but I think look, what I seem to see is there's a growing sort of body of evidence to suggest that there's a place for ctDNA sequencing at regular intervals, even in someone who is probably responding quite well to treatment. Just seeing if that's the case, and that's what this sort of study sort of pointed to, and if that is also the case, how would that potentially change someone's treatment path that was already on a 3rd generation TKI? So let's say, for instance, they were, we're doing brain MRIs every 6 months now. Would you then throw in conjunction with that ctDNA just to pick up any sort of tumor loading that might be increasing in the blood or liquid biopsy? And if so, then what would you do if you're already on 3rd gen TKI?
Yeah, exactly. Because you're right. Right now, when the drug is working, what the blood shows doesn't change the treatment. So right now, the results and generating more of these types of studies are important to inform, the research we do with treatment. And we have to make sure that that's obviously, very strictly designed. So an example, as I said earlier, if you still had circulating tumor DNA on lorlatinib, there may be an important study to do where we looked at adding some chemotherapy. Again, this is something that isn't mainstream, but these types of studies and showing the need in certain individuals who may have some changes, like, extra different mutations, informs, well, like we saw in ALKTERNATE, they need more. They need something more than this. And so that's how we take that forward with a study. But what's super important, though, is that we do that in those formal channels, because although we may have a hunch and a bias, we may also be wrong. So that's also why the way we do it needs to be very scientific, so that we're making sure that it's being shared with the scientific community, published, not anecdotes. So right now, as I said, we keep monitoring. So perhaps if I see that, though, I might monitor a little bit more closely. If there were some risk factors, I might be starting to think if there's something on the PET that is still, maybe lighting up, I might think, hang on, what's going on there? Do we have a discussion with the MDT about whether we do some radiotherapy to that site? Of course, there are ways we try and do some extra, personal things. But to say right now, based only on the blood, you do something differently when the cancer's under control, it's too soon.
Yeah, understood. Yeah, thanks a lot.
Excellent. Look, before we say thank you to Malinda for her time, in the meeting invite, I attached what Malinda described as a lay summary of her journal article. I think this is a great approach that is being taken to try and take some of these complicated journal articles and put them into a bit of a language that's a bit more accessible, particularly to patients. Malinda would love a bit of feedback on that article if you're able to provide it. Please feel free to send those comments through to my email address, and I'll collate all of that and summarize it and provide it back to Malinda so that she can try and improve that summary. Have a read, see if you understand it, see if you've got anything to add to it. But please take the time with me to thank Malinda to share that trial with us. She's an exceptional oncologist, and I really appreciate the approach that she takes to working with consumers like ourselves and patients and the people who care for us. She's one of the most accessible oncologists out there, and a lovely person as well. So thank you very much, Malinda. It's really appreciate the time that you've taken for us. Go and rest up now and say hello to your kids.
No, they're sleeping. And by the way, I feel the same in the other direction. Thank you for having me. And I love working with you as well. So it's very easy.
It's a pleasure.
But yes, everybody needs to hopefully, yes, be in bed soon. So thank you and be in touch. And obviously happy for emails if there's any questions after tonight.
Thank you very much, Malinda. Much appreciated.
Lovely to see you all. Bye.
Bye.